Okay, we're going to continue on with the respiratory system now, and we're going to get into the concept of gas exchange. So first, let's talk about gases. We've already discussed Boyle's law, so the idea that volume changes lead to pressure changes, which lead to gases flowing down their equilibrium. So that is going to play into this. Dalton's law of partial pressure states that when gases are in a mixture, each gas exerts its own pressure, which is a partial pressure of the gas as a whole, of the mixture. So air is made up of 4 or 5 different gases. Each one is going to exert its own pressure to make up the 760 millimeters of mercury that we see when we talk about atmospheric pressure. Henry's law is going to come into play when we're talking about that layer of water that our gases are going to have to diffuse across. So Henry's law states that when gas is in contact with a liquid, the gas will dissolve in the liquid in a proportion to its partial pressure. So the more of something that is in the mixture, the more of that will dissolve. And the molecules go back and forth between the dissolved and gaseous state until equilibrium is reached. In terms of moving through this fluid, solubility is definitely going to come into play. So something like oxygen is not terribly soluble. It does not dissolve in water very well. CO2 has a much higher solubility, and so it's going to be a lot easier for CO2 to diffuse through this layer. And then temperature will also play a role. As temperature increases, solubility decreases. So these are some of the properties of gases that we're going to be looking at. So if we look at air, we basically have 4 major components here. So it's primarily nitrogen. So air is going to be somewhere in the neighborhood of close to 80% nitrogen. And so the partial pressure of nitrogen is about 597. So basically, if you take 78.6% of 760, you're going to get 597. That's how you calculate the partial pressures. Oxygen makes up about 21, so a little bit of water and a small amount of CO2. So this is basically the air that we breathe. If we compare that to the air in the alveoli, we see that nitrogen doesn't change that much. We're not going to deal too much with nitrogen. Oxygen goes down significantly, though. So it goes from about 21 down to about 13. Water increases significantly, and so does CO2. So there's a difference in the composition between the air that we breathe in and the air that is in the lungs, and we can see exactly why this is happening. First of all, gas exchange. So the oxygen and CO2, that is the result of gas exchange happening. The reason that we have such an increase in water is due to humidification, so that process of adding moisture into the air. Now, down in the alveoli, in that residual volume, there's always a mixture of new and old air. So, like I said, it takes about 90 seconds to fully refresh your residual volume. So you're always mixing old and new. All right, so let's talk about gas exchange. So we'll start in the lungs. So during pulmonary gas exchange, oxygen is moving into the blood, CO2 is moving into the alveoli. We're going to have a few different factors that are going to come into play here in terms of how well this is going to work. First comes the respiratory membrane. So we talked about this when we were looking at the alveoli, and we said that we've got the simple squamous cells of the alveoli, a basement membrane, and then the endothelial cell of the capillary. So that is your respiratory membrane. Anything that increases the thickness or decreases the surface area of respiratory membrane is going to negatively impact gas exchange. So we're going to talk about a couple things that would do that in a second. Next thing would be the partial pressure gradients and the solubilities of the gases. For oxygen, we have a very steep gradient. And that steep gradient is going to allow oxygen to overcome its lack of solubility. The gradient for CO2 is much, much lower because it's more soluble. So we don't need to have such a heavy, steep gradient if the gas is already able to very easily dissolve in water. So we see two differences there when we're looking at the gradient versus solubility. The last thing is a concept called ventilation-perfusion coupling. And we're going to talk about this in a second, but these two things have to be matched for gas exchange to occur efficiently. So all of this is happening up in the lungs. When we get down to the tissues and we move into internal respiration, we're going to reverse this, but there is not quite as many factors coming into play. So let's talk about these concepts here. So first, let's talk about the respiratory membrane. Here is what a normal histological slide of the lung would look like. If we have pneumonia, we're going to collect fluid in the lungs, and the respiratory membrane is going to be thickened. That is going to hinder gas exchange. Anything that thickens up that membrane is going to significantly decrease our ability to oxygenate our blood. Emphysema destroys the surface area. So we have giant alveoli because the walls between them are destroyed. Here we're losing surface area. Also is going to negatively impact our gas exchange. So anything that increases thickness or decreases surface area is going to have a negative impact on gas exchange. Here we're looking at this concept of the steepness of a gradient. So this would be like the normal oxygen gradient, okay, that's going to occur, and it's much, much steeper than what we see for CO2 because we're just not able to get oxygen to dissolve as easily in the water. Now, the steeper the gradient, the more oxygen diffusion. So if you're in a hyperbaric chamber, you have a very, very steep gradient, and it's going to force oxygen into the tissue, which is exactly what we would want to do in that case. The last thing is the concept of ventilation-perfusion coupling. So what this means is that you need to make sure that the amount of blood and the amount of air match. So for example, here we're looking at what would happen if we have a decreased airflow. So decreased airflow means, okay, I need to decrease the amount of blood that's going to this area so that the ventilation and the perfusion match up. So it would cause vasoconstriction of the vessels, which would decrease blood flow, and we would end up matching our ventilation. On the flip side, if we increase airflow, we have an increased oxygen level, we're going to vasodilate, it's going to increase the blood flow. Now we're going to bring in more blood to oxygenate because we've got more air. Okay, so this is an example of how perfusion can adjust to changes in airflow. The opposite can also occur where we've got a decreased blood flow. So we have a decreased CO2 here, constriction of the bronchioles, decrease the airflow, and we are going to match what's happening with the blood. On the flip side, if we increase the blood flow, we're going to dilate the bronchioles and increase the air. So you want to think about, when you're talking about ventilation-perfusion coupling, it's all about making sure that the amount of air matches the amount of blood. If you're bringing air in, but you don't have enough blood to pick up the oxygen, that's not efficient. If you have too much blood and not enough air, then the blood's going to leave not having picked up its oxygen. So ventilation and perfusion need to match, and either one, the blood flow or the airflow, can adjust to the other. Okay, so now we're going to talk about the actual gases, starting with oxygen because it's very simple. When oxygen is being transported in the blood, there is a very, very small amount dissolved in the plasma. Again, it's not terribly soluble. The majority of it is bound to hemoglobin. And that's going to form what's called oxyhemoglobin. As soon as the first oxygen binds, it's going to increase the affinity for the other three, just as the first oxygen that disassociates decreases the affinity for the other three. So once you have one bound, it's more likely to have all of them bound. Once you get rid of one, it's easier to get rid of the others. Now, this is a reversible binding, and there's several factors that are going to play into how much oxygen disassociates from hemoglobin out in the tissues. So we're going to look at 5 different things. The first is the partial pressure of oxygen. If the tissue has a low level of oxygen, it's going to increase dissociation so that the oxygen can move off the hemoglobin and into the tissues. If temperature increases, it's going to cause an increase in dissociation, usually because as temperature goes up, it means the tissue is being more active, it means it needs more oxygen. As blood pH decreases, which remember means we're gaining hydrogen, it's going to cause an increase in dissociation. That usually goes hand in hand with the next one, which is an increase in CO2. The more CO2 I have, the more oxygen I need to disassociate to get us back into balance between oxygen and carbon dioxide. And then finally is BPG, bisphosphoglycerate. This is a byproduct of glycolysis, which is anaerobic respiration, meaning that these cells are not utilizing oxygen because they don't have it. So the more BPG that builds up in a cell, the more oxygen starved the cell is, which means we're going to increase dissociation. Oftentimes all of this is happening at the same time. So temperature goes up, oxygen levels drop because we're being very active and the temperature is going up. And as oxygen drops, CO2 increases. The more CO2 increases, the more our blood pH drops. And all of this is going to require anaerobic respiration if we are in a cell that could undergo it. So oftentimes all of these happen together and they are all related to each other. So here we can kind of look, if you're just hanging out, laying in the grass like this girl here, her hemoglobin is 75% saturated. So only one of the 4 is being released. But if we're over here and we're really working the tissues and we would think, okay, we look at this skeletal muscle here. It's working really hard. Temperature's going up. Oxygen's going down. It's probably collecting BPG. CO2 is going up. pH is dropping. All of those situations are going to drive more hemoglobin or more oxygen to disassociate from the hemoglobin. So his saturation's down at 40%. So it's going to be the physiological state of the tissue that's going to dictate how much oxygen is going to be released from the hemoglobin. Now, when tissues don't have enough oxygen, they become what's called hypoxic. So it just means that we're not getting enough oxygen to the tissues for some reason. There are multiple reasons. So the first would be an anemic hypoxia, meaning that you don't have enough red blood cells or your hemoglobin is abnormal and so it's not carrying oxygen the way it should. So this we need to treat the anemia so that we can relieve the hypoxic conditions. Ischemic means that there is a block in blood flow. So the tissue is becoming ischemic because there's either an occlusion in the blood vessel or there has been some type of damage and the blood is not getting there. So the blood is probably oxygenated at a normal level, but it can't get to the tissue. So when you have a blocked coronary artery and the muscle cells downstream from the blockage become ischemic, it's because the blood can't get there. It's not because there's no oxygen in the blood, it's because the blood can't get there. Histotoxic means that you've got normal levels of oxygen and normal delivery to the cells, but the cells can't use it. So there are different types of toxins, things like cyanide, that make it impossible for your cells to actually utilize the oxygen that's being delivered to make ATP. And then finally, we've got hypoxemic hypoxia. this is an actual reduced arterial oxygen level. So hypoxemia means that the issue is likely in the lungs, where we are hindering gas exchange for some reason. One of the things that can cause hypoxemic conditions that isn't related to damage in the lungs is carbon monoxide poisoning. So we have a lot of carbon dioxide in our body, and when we look at the transport of oxygen and carbon dioxide, they're not competing for a binding spot on the hemoglobin. Oxygen is going to be carried by the heme group, and the CO2 is going to bind to the globin portion. Carbon monoxide does compete with oxygen for the binding site. And in fact, the affinity for carbon monoxide is higher than the affinity for oxygen, which means that in the presence of carbon monoxide, oxygen will get pulled off of the hemoglobin and will be replaced with carbon monoxide. So when you're dealing with somebody who has carbon monoxide poisoning, they will present as hypoxemic. They will have a much lower than normal oxygen level in their blood. All right, carbon dioxide transport is a little bit different, a little bit more complex. First of all, we're going to dissolve more of it in the plasma. So it's more soluble, so up to 10% of it can be simply dissolved in the plasma. About another 20% is bound to the hemoglobin, again, to the globin. Okay, it forms carbamine hemoglobin. But the majority of it is in the form of a bicarbonate ion. So this equation right here is a very important equation reaction in the human body. You're going to need to understand it now in the urinary system, in the homeostatic system, in the digestive system. It's just going to keep coming up over and over and over again because we have a lot of both reactants. We've got a lot of CO2 in the body, and we've got a lot of water in the body. When these two react, they form carbonic acid. And then carbonic acid can further dissociate into a hydrogen ion and a bicarbonate ion. This right here, this is all reversible, so it goes in both directions based on how much of either end we have, okay? So when we looked at the factors dealing with oxygen dissociation, we said that high levels of CO2 generally cause decreases in pH. The more CO2 I have, the more I drive this equation in the forward direction, leading to the formation of hydrogen ions that drop my pH. So there is a significant relationship between the amount of CO2 in the body and the pH of the body. This is why right here, the changes in respiratory depth and rate affect blood pH. Remember we said that pH was the single most important factor for changing your respiratory rates? And this is why. Because your respiratory system is the most important factor in determining how much CO2 you have in your body. How much CO2 you have in your body is directly related to how much free hydrogen ion you have in your body. If you don't have a lot of CO2, you're going to draw this equation in the backwards direction, and you're going to bind up this hydrogen in this water, and it's not going to be affecting pH anymore. Okay, it's not going to be dropping our pH. So when we talk about this relationship between CO2 and pH, it is right here, and it's going to come back over and over and over again. This reaction will happen on its own, slowly, but when we want it to happen really quickly, we're going to find this enzyme, carbonic anhydrase. Anywhere we find carbonic anhydrase, this is going to happen very, very quickly. Opposite direction as well. So we're talking about CO2 transport. So we're saying, hey, we loaded on a bunch of CO2 at the tissues, and some of it got dissolved in the plasma, a little bit more bound to the hemoglobin, and the rest of it, up to about 70%, is traveling as a bicarbonate ion. So this CO2 moves over here and is bound up in a bicarb ion. That is then going to get moved out of the red blood cell using something called the chloride shift, where it's going to become part of a buffer system. So the carbonic acid, so from here over, the carbonic acid bicarbonate buffer system is the most important buffer you have in the extracellular fluid, including your plasma. So essentially what's happening with this CO2 molecule that we picked up at the tissues is we're making it work for us on the way back to the lungs. We're going to push it into this buffer system, and then when we get back to the lungs, we'll reverse all of this so that we can exhale some of our CO2. But the most useful place for the CO2 to go for us is into this buffer system. So let's take a look at this. So here we are in the tissues, okay? So in the tissues, my job is to move oxygen from the blood into the tissues and move CO2 from the tissues into the blood. So let's look at this, okay? So here I've got my hemoglobin that's bound up to oxygen, okay? And in order to drop off most of this oxygen, what I do is I bring in a hydrogen ion that binds to the hemoglobin and kind of kicks the oxygen out. It helps release the oxygen. So the majority of my oxygen is going to be just released simply from the hemoglobin and head into the tissue due to the gradient present for the partial pressure. The small amount that's dissolved in the plasma simply diffuses on its own. So there we've taken care of moving the oxygen out. Now what do we do with the CO2? Some of it is going to move in and dissolve in the plasma. Some of it's going to move in and it's going to bind to plasma proteins and be hanging out with them in the plasma. And some of it's going to move into the red blood cell and bind to hemoglobin, forming carbaminohemoglobin. The majority of it, though, is going to move into the red blood cell, and in the presence of carbonic anhydrase, is going to form carbonic acid, which is then going to dissociate into bicarbonate and a hydrogen ion. Now, the bicarb isn't doing us a lot of good in the red blood cell. We want it out here in the plasma where we are able to make it work for us in the buffer system. So we use an antiporter. It's called chloride shifting. We move bicarb out and chloride in. They're each negatively charged ions, so we can exchange them with each other without messing with tonicity at all. We're going to see chloride shifting multiple times. So when we move bicarb in one direction, we bring chloride in the opposite direction so that we can maintain the proper charges. And then this hydrogen that we made is what actually comes down and binds to our now empty hemoglobin. So all of these pieces are now taken care of, and we travel toward the lungs. When we get to the lungs, everything needs to reverse. So the oxygen comes in, dissolves in the plasma. Most of it comes in, and in the presence of this massive influx of oxygen, it's going to pop that hydrogen off of the hemoglobin. So I'm going to get oxyhemoglobin again. And that hydrogen is now going to come up here. We're going to reverse the antiporter. We're going to bring bicarb in and take chloride back out. The hydrogen and bicarb are going to force the equation in this direction, and we're going to make water and CO2, and this CO2 diffuses out down the partial gradient. The carbaminohemoglobin is going to release. We're going to move out into the alveolar air. The plasma proteins that were carrying our CO2, they're going to move out, and finally the dissolved is going to move out. Now, the movement and why they move are the partial pressure gradients, the fact that there is less CO2 here than here. There is more oxygen here than here. All of these equations, they simply work in one direction at the tissues and in the opposite direction in the lungs. And then when we pick it up in the lungs, We're then going to head down to the tissues, and we go through all of that again. So all of this is going back and forth constantly as we go through this concept of the gas exchange. So this is something that you kind of need to work on a little bit. You need to kind of draw out these pathways to give yourself a better view of kind of how all of this works. And none of it's terribly difficult. It's just a lot of sort of chemistry all at one time, but it's just the same equations going in one direction or the other based on the gradients.